So, we've been talking about sex differences and how they come about. And what we've seen so far is that in mammals, at least, the presence of testosterone or other androgens early in life means that sexual differentiation of the nervous system is altered. And for the most part, what's happening is testosterone alters the rate of cell death or apoptosis. So that in the SDN-POA, for example, the presence of testosterone early in life keeps neurons from dying in the SDN-POA, and that same testosterone early in life promotes the dying of cells in the AVPV, so that by the time you get to adulthood, males have more neurons in the SDN-POA than females do, while females have more neurons in the AVPV than males do. And again, for the spinal nucleus of the bulbar cavernosus, testosterone early in life keeps the muscle target muscles from dying, the bulbar cavernosus muscles. And once the muscles are kept alive, then the motor neurons are kept alive. And since that doesn't happen in females, then you end up in adulthood where males have more motor neurons in that nucleus in the spinal cord, the SNB, than do females. But there's also evidence that androgen acting early in life can also affect differentiation. And the cutting edge for this chapter is concerning that, where the individuals did a single cell analysis of gene expression in the bed nucleus of the stria terminalis, where there's known that there's a sex difference. And here we're looking at the bed nucleus of the stria terminalis just generally, and you see that these are all neurons. That you don't see that, but these are all neurons, and you note that there are several different types of neurons. And in fact, these aren't just neurons. These are all neurons that are GABAergic. So they are cataloging 7 different types of GABAergic neurons that are found in this region. And here they are taking that same data, only now instead of color coding it to let you know what type of GABA neuron it is, here their coding is just either female or male. And you can see that there are some areas, some cells that are expressing patterns of gene expression in males but not females, and other cells are being found more other patterns of gene expression are more common in females than males. So here's an area where these are mostly males that are expressing genes in exactly this way in their GABAergic neurons, and here females, and so on. And so to test whether this sex difference that they could document was due to differences of exposure to testosterone early in life, they then did another experiment where they took a look at the proportion of BNST neurons and kept track of them that way. And indeed, there were, for these 3 different types of GABAergic neurons, type 1, 2, and 3, they could see that there were more of these cells being expressed, more of the pattern gene expression in males and females for 2 of the GABAergic cells, but females were expressing more of another type of GABAergic neuron, whereas for most of the GABAergic neurons, there was no sex difference. And so they concluded that one of the things that early androgens can do is affect the gene expression in GABAergic cells in the BNST, and in that way cause more cells to take on the type 1 GABAergic phenotype in males and females, and more to take on the type 3 phenotype in males and females, whereas in type 2 GABAergic neurons are going to be more common in females than in males. So when we get back to class, that will conclude the questions about hormones. And when we get back, we'll have a quiz about that, a Kahoot. But now I want to get to the final part of the chapter where it doesn't actually involve hormones, but it does involve sexual differentiation. And it's sort of included here to show you how things could have been very different. So in flies, unlike mammals, Sexual differentiation has nothing to do with hormones. In fact, flies don't have any androgens or estrogens for that matter, or to the extent they have estrogen-like compounds, they're just generally used for nurturing eggs and embryos and the like. Rather, in insects, sex differentiation is a purely cell-autonomous effect. That is, every cell in the fly's body will take on either a male or a female phenotype based on genotype alone. And the rules in flies are a little different than as in mammals. In flies, XX individuals typically become female, and XY individuals typically become males. I'll warn you, I mentioned this before, this pattern of sexual differentiation by chromosomes evolutionarily has nothing to do with the pattern of chromosomal sexual differentiation seen in mammals. These were two independent events that arose during evolution. And in fact, there have been several times where the idea of having males or females determined by which chromosome they get have arisen independently. And there are some vertebrate species in which chromosomes do not have an effect on sexual differentiation. That is, there's some in which, for many lizards and reptiles, for example, it's not what chromosomes are inherited that determine sex, but rather the temperature at which the eggs are incubated. And in any one big clutch of eggs, some cells will be just above threshold for making one sex, and others will be others. And it works out such that the typical sex ratio that's produced is about 50-50, depending on where exactly the egg was in the mound. And just as an aside, one of the things that climate change is threatening is you can imagine a situation where if climate changes enough, that every egg will come out either male or female. And once that happens, if all the clutches are hatching only one sex, of course, the population is doomed. Anyway, to get back to sexual differentiation in flies, where it is chromosomal, but it's a cell-autonomous response. In flies, in insects, I should say, no, it's specific to flies. Different insects have different, in fact, chromosomal mechanisms for controlling sex. But in flies, 2X chromosomes indeed means that there will be females, but and we're going to talk about that. And it's the presence of 2X chromosomes that means that there's going to be more transcription factors for a particular transcription of a particular gene that is called sex lethal. And the reason it's called that is when it's not functioning, one sex carrying it will always die before hatching. But we're not going to concern ourselves with that. The important thing for us about sex lethal is, despite the misleading name, is it's going to control the sex of the rest of the animal. So this sex lethal protein regulates mRNA splicing, and one of the first things it does is make sure there's going to be more and more sex lethal protein being produced. Since females have 2X chromosomes that cause this to happen, that means they'll have a threshold amount of the sex lethal being expressed early in development, and that means the genes will continue to express more of the sex lethal gene throughout the life of the animal. So one of the other things that the sex lethal protein does is trigger a whole bunch of feminine gene expression, and again, that effect is happening in every individual cell. One of the things this means is that, unlike the case in mammals, where an XO individual will make ovaries and therefore make a female phenotype, in flies, an XO animal, when there's only one X chromosome and no second sex chromosome, in those animals, because there's not two copies of the sex lethal gene on two different sex X chromosomes, in those animals, there won't be a sufficient amount of the sex lethal protein expressed early in life to upregulate its expression the rest of life, and therefore those animals will look like males. And one of the consequences of this cell-autonomous sexual differentiation is that it occasionally happens where an X chromosome gets lost at an early stage of development, say when there's only going to be 2 cells that are going to give rise to the entire fly. And when that happens, what you end up with is in a situation where half of the animal is female and the other half is male. And such individuals are known as gynandromorphs. So they are both female and male. And so in flies, this was established by, as you might have expected, by looking at different mutations on the X chromosomes and showing that in an XX female, when there is a wild type copy of the good genes, that you have a wild type phenotype, whereas in the other half of the animal, where there's only one X chromosome, then these mutations express so that you have an eye that's the wrong color and a wing that's smaller. It also sometimes happens, this was done deliberately, but it sometimes happens spontaneously. And so butterfly collectors get really excited when they find a butterfly in which males and females typically have very different morphology. Well, it sometimes happens that an X chromosome will get lost at that early stage, so that you'll end up with an animal where half the animal has a female phenotype and the other half has a male phenotype. And these two are known as gynandromorphs. And so that's a brilliant demonstration that in flies, sexual differentiation is a cell autonomous response. Each cell in the entire body, including the nervous system, is going to take on either a male or female phenotype, depending on whether it has 2 X chromosomes. Among the other things that are involved in sexual differentiation in flies is courtship behavior. So here's the normal courtship behavior in Drosophila. As in many mammalian species, it's the males who are doing most of the courtship, who are trying to persuade a female to give them some sperm. So the male will first find a female based on visual cues, and then he'll start taking his leg to tap her body, and he will at the same time extend one wing, either the left or the right, and give a little buzz that can be considered a courtship song. And if she stands still for that long enough and cooperates in other ways, then he'll start licking her genitalia. And if she lets him get that far, she may then allow him to copulate. And at that point, sperm transfer happens, and there are more flies on their way. Well, this courtship behavior is, of course, very much influenced by genes in flies, and there was a very interesting phenotype that was seen in a gene where the first mutants of the gene eventually got to be called Fruitless. You'll see why in a moment. And the first mutants of Fruitless were males, but they would not court females, or they would court only males. And so you'd end up with a situation where there'd be chains of males, each investigating the rear end of the other, and all of them trying to court one another. And so it was clear that Fruitless, the gene, normally played an important role in male courtship behavior. And as they came up with new mutations of the Fruitless gene, one of the things they eventually found out was that the genetic sex, whether 2 X chromosomes, and early in life, whether there are twice as many transcripts for the sex-lethal gene that we talked about before, that the genetic sex will determine which of 2 different transcripts will be made from the Fruitless gene. So there are some transcripts that are normally made only in females, and other transcripts that are normally made only in males. And whether they express one another, whether they express the male transcripts or female transcripts, depends on the cell autonomous sex of the cell, as determined by sex lethal, etc. So XX individuals make fruit transcripts that are non-functional. This is kind of an interesting thing. They make transcripts, the genes are being transcribed, but no protein ever gets translated out of it. On the other hand, XY individuals make 3 different functional transcripts, and these male-specific transcripts are important for courtship behavior. So one of the ways to test this is they made constructs that express different Fru transcripts in flies and they put this on a background in which the fruitless gene was otherwise knocked out. And what they found is that if they made, let's see, so here's males courting other females, right? So in wild-type animals that are carrying the wild-type alleles for Fru, they are courting exclusively exclusively females. And male flies carrying the wild-type allele or a replacement copy that can produce all male transcripts, they courted females as normal. But males that could only produce female typical transcripts of Fru did not court, and those producing male typical transcripts did. So you see this? They take flies in which the fruitless gene itself is knocked out, and then they make constructs that are where they're either expressing the female version of the transcript or the male version of the transcript, and only if the male version of the transcript is present will these males actually court females. On the other hand, in terms of female-female transcript, normally females who are expressing the normal gene for fruitless, and and therefore expressing only females specific transcripts, or if they're missing the fruitless gene and they've been transfected so that they're making the female transcripts for fruitless, they don't court other females. But put into a female transcripts for the male-specific transcript for fruitless, and lo and behold, when they grow up, they'll be just about as interested in courting females as males were. And so here's an example of one of these construct females. So her endogenous fruitless gene is knocked out, but she's been manipulated so that she's expressing male-specific transcripts for fruitless. And here she is courting another female with her wings sticking out, ready to sing to it. The other thing that's fascinating about this, and I'm not going to go over these things, these experiments, but they eventually showed that the important place for the fruitless male-specific transcripts to be expressed, as you might expect, was in the brain. And when they cataloged protein expression in a male brain, they noticed that there's a lot more of this fruitless protein being expressed. This is all the brain cells in various neurons scattered around the brain in males, and in females, you don't see it as much. And what they could also do is then define a fruitless gene-expressing network in the male brain, and by labeling the same neurons, they could see that the morphology of that network of neurons, they're not expressing these are the ones that are expressing the Fru gene, but not making the Fru protein. That same bunch of neurons has a very different expression pattern in females. And remember, the female-specific transcripts don't make protein, and that's why when you're looking for the protein in the female brain, there's none to be had. But we can still identify the female homologue of that network by keeping track of whether they're expressing the gene, even if it doesn't make any protein. And there's a big sexual dimorphism here in these networks. Well, all right, so the whole point is that sexual differentiation could have been a cell-autonomous response to just gene chromosome inheritance in our species and other mammals, but it's not. It's very much hormonally dependent. Then the last thing I'll say about sexual differentiation in flies is interesting because it's somewhat it's reminiscent of the SNB in mammals. So flies have these muscles in the abdomen that are known as the muscles of Lawrence, named after the guy, of course, who first noticed them. And the interesting thing about these huge abdominal muscles is they're only found in males, and you won't be surprised to learn that they're helpful in terms of the male bending over to get his abdomen there to to copulate with females. Anyway, these muscles are only present in males. Females don't have them. But, and again, genetic sex is important for causing the sex difference, but in some ways, the rules are different for the SNB. Remember, for the SNB, the hormone is involved, not genes directly. The hormone keeps the muscle alive, and the muscles therefore keep the motor neurons alive. So in that case, the target muscles first are sexually differentiated, and then they cause the motor neurons to be sexually differentiated. For the muscles of Lawrence in flies, two things are different. Number one, hormones have nothing to do with it. The question is, what chromosomes do the cells have? And it turns out that in that case, if the motor neurons have the sex chromosome complement for males, if they're masculine, they will then keep the muscles alive. So in that case, the differentiation factor is genes, not hormones, but the genes are not acting on both of these tissues in a cell-autonomous fashion. Rather, the gene is acting in the motor neurons, and as long as the motor neurons are male in genetic sex, they will keep the muscles alive, and the muscles will have a male-specific phenotype. So this is, interestingly, this is an exception in the case that you can end up with an animal in which the muscles are XO, and yet they're still masculine because they're dependent on the motor neurons to determine whether they're going to be around, whether they're going to survive, and whether they're going to take on a male phenotype, being nice and big. Anyway, so I wanted to finish up that chapter before we meet on Thursday. And so I wanted to go over that material. When we come back on Thursday, you won't be surprised to learn that we'll have a Kahoot right away. And so if you haven't already, I'll remind you that the reason we're recording this lecture is so that to make it easier for you to vote. And so if you haven't voted yet, I will urge you to get out there and vote for the candidate of your choice, whoever that may be. Anyway, so hope to see you all on Thursday.